This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on February 28th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we are in Bethesda, Maryland. We're on the campus of the National Institutes of Health, and joining me today here, Rich Condit.
Hello, Vincent. On the road again. This is great.
Second day here in Bethesda.
Yeah, and I neglected to look up the weather, but it's cold.
And our guest is an investigator at the NIH. He's an NIH Distinguished Investigator. He's the chief of the Genetic Engineering section. And of course, this is NIAID, National Institute of Allergy and Infectious Diseases, part of NIH. Bernie Moss, welcome to TWiV.
Welcome to NIH.
Thank you. And you've been here for 50 years, right?
54.
Wow. That's amazing. That's amazing. That's a long time. I'm at Columbia for, 39 years. I thought that was a long time, but it's not even close. I'm not going to make 50.
I don't think I'm the longest. I think there's a few that have been here longer than I have.
Wow.
Really? Wow.
Just a few.
So this building is where your lab is, right? So we're in a conference room here, and we're grateful that you took the time out to talk with us today. Somebody we wanted to talk to for many years.
For a long time. We've been talking about this for a long time. We finally got it together to make the trip. We've actually made a special trip here to do these interviews.
These guys. Now, this is the second of 3 TWiVs we're going to do here. They'll be released over time, so me saying that there are 3 won't make much sense, but don't worry about that. Of course, Bernie is special to Rich because you guys both work on poxviruses, and so in particular for you, but we all hear about poxviruses all the time. So Bernie, let's start at the very beginning. I can tell by your accent that you're from somewhere in New York, right?
That's right, from Brooklyn. I think my accent is getting stronger with the years,
Really?
Actually.
Fred Murphy is from Queens, but he has no accent left because he's moved around. He's been in Texas and Georgia and California. He's kind of lost it. Mm-hmm. But I guess since we're so close to New York here, you never lost yours.
Well, whatever.
So you were born and raised in Brooklyn?
Correct.
And you're first generation. Is that correct? Your parents were immigrants?
That's right. My parents were immigrants. On the maternal side, they came soon after World War I. Uh-huh. So my mother was about 11 years old.
What country from?
It's hard to say. Do you mean what country after the World War or what country now?
Yes.
I think the Ukraine is where it is now.
Okay.
But it was Poland then.
Oh, that's interesting.
Interesting.
And your father as well, the same country?
No, he came from Romania.
Okay.
He was actually born in the UK because they were en route to the United States.
Wow.
Interesting.
How old was he at the time when he emigrated?
He was born in the UK.
Oh, okay.
Yeah, UK.
So I think they spent a couple of years there.
Okay.
Yeah.
So, yeah.
You speak any of those languages?
No.
Okay. So you grew up in Brooklyn, and your parents, what did they do?
So my mother was a housewife.
Right.
And my father was in retail stores.
Stores. So there was no science in your family, right?
No, there was no science and no university.
Did you have siblings?
But they encouraged me.
Yeah, to go to college.
To go to college, especially to become a medical doctor, because in the immigrant community, that is, a path to independence and security.
Yeah.
So I started out following that pathway. But when I got to college, and especially a cell biology course.
Which college?
I took NYU.
Ah.
Yeah, we didn't. So our horizon for higher education didn't go beyond New York City.
But NYU was a good school, right?
Yeah.
Yeah.
Still
So, so the cell biology course, Paul Gross was the lecturer, and that really fascinated me. And I took an honors program, so I did research in his lab. And I thought about switching to a PhD program, but I had already been accepted to medical school, to NYU. And they actually had a pretty strong science there. So I decided to continue. And
so you went from Washington Square to...
I was in the Bronx. There was a university college at the time. It's no longer open.
Oh, I didn't know that.
Yeah.
And then the medical school, of course, is on the east side near Bellevue, right?
Right.
Okay. We should say, by the way, that all this is in your autobiography in Journal of Biological Chemistry, which I read at the Starbucks this morning.
We'll put a link to that in the show notes.
So it's fresh in my mind. And so the medical school at the time had some interesting faculty, right?
That's right. Severo Ochoa won the Nobel Prize while I was there.
Wow.
And it was good. It's a good place.
So you write in your autobiography, you enjoyed the biochemistry course, which was attended by other members of the department.
Yeah, Severo was really a very strict administrator. Everybody in the department had to sit in the front row and listen to every lecture so that there would be continuity.
Good for him.
We were reminiscing, Rich. Today, nobody does that anymore. It's a shame in a way.
It's a shame.
I'm not sure even the students attend.
That's right.
Good point. A lot of online teaching and that kind of stuff.
So another famous graduate of NYU, and many, is Jonas Salk, which is many years earlier, though, like in the 40s or something like that. Yeah. All right. So you're in medical school, and you're getting some research experience there? Or?
Yeah, I had 2 elective periods, and I did it in the biochemistry department. Professor Robert Chambers, and he was a real biochemist, really an organic chemist almost. So it was very good training. And he was, he looked over my shoulder all the time, and I had to keep a notebook, and it was a great way to learn. I had to write every single thing down. If I weighed something, on the left side of the paper, I would have to put down exactly the numbers I had. And on the right side of the page, the experiments. So that if anything went wrong, he could open it up and say, "You dummy, you didn't weigh it right."
Wow. So
that was really good training.
Yeah, sure.
Yeah, I remember one of my first advisors training me on note taking. Okay, and you know, I was okay at it. Okay, and I tried to train my students as well, but so many times I would look back months later at an experiment or something like that and say, I wish I'd written that down. Something that I didn't keep good track of.
Yeah.
It's a good thing to learn.
It's an important skill.
So one of the experiments I had to do, we were working with pseudouridine, and the source of pseudouridine was urine. So I had to set up large bottles in the men's bathrooms to collect it all and to bring it back to the lab. And then my first task was to extract the nucleotides from that.
Great.
But the work went well, and I actually published a paper on the synthesis of nucleoside triphosphates.
I think most importantly, the biochemical training is what we see in the rest of your career, right?
Yeah.
That you got back then. If you had done something else, you might have had a different interest.
maybe.
Yeah, you never know. Okay, what did you do after medical school?
Okay, so after medical school, I decided to take an internship.
Mm-hmm.
And I initially I was starting to apply for medical internship.
Right.
And then in my last year, I took medicine in the Bellevue wards.
Mm.
And I quickly decided to change to pediatrics.
So at this point, when you've finished medical school, did you still envision yourself as a practicing physician down the road?
No, I didn't.
Right.
No,
Okay.
I knew I'd do research, and it was just the cap on the medical education.
Okay.
To take an internship, and then I was finished.
Okay. Mm. You write in your autobiography that the Bellevue was tough, right?
Bellevue was a good experience because of the shortage and the staff.
Yeah.
So the medical students did things that an intern would probably do in other hospitals.
Mm. Wow.
Whether that's good for the patients is another story.
Yes. Good for the students. All right. So you do an internship. Where was that?
The internship was at Children's Hospital in Boston. And it was my first choice because there was great research coming out of there. Mm-hmm. If you remember Enders,
yeah.
He was the first to grow a virus in tissue culture. It was poliovirus. But of course, as an intern, I never saw Enders or any of the faculty except for the clinical faculty.
So you knew about the Enders experiment at that point,
sure.
Right? Everyone knew it, I guess. It was a big deal. Interesting. Yeah. Nowadays, nobody knows it anymore, right? It's all lost. But I do teach it. So you mainly spent that year taking care of patients, right?
Completely taking care of patients.
But it was your intent that would be it, and then you'd go on to research, right?
Yeah,
okay.
Yeah.
So, and that was at a time when there were no restrictions on how long interns should work in a day or something like that, right? So you were pretty busy.
We were we were very busy. It was every day and every other night and no holidays.
Right.
To get a break in the for Christmas, for example, we informally worked every night for a week and then switched with somebody.
Wow.
That was really tough. Yeah.
So you go ahead, Rich.
Then what comes after the internship?
Okay, so after the internship, I wanted to get research experience. And I felt that my basic science was not strong enough. So I decided to take a PhD rather than a postdoc so I could have a year. And that was facilitated by a grant that I got that was administered by NIH, and it was specifically for training physicians. But with that fellowship in hand, I could go any place. And so I went to MIT.
What year would that have been?
That was 62.
Okay.
And you mentioned that Lewis Thomas played some role in you getting this fellowship.
That's right. So Lewis Thomas, I don't know if your listeners know about him, but he was a very literary physician, and he wrote a weekly article for the New England Journal of Medicine, and he wrote a lot of books. Notes of a Biology Watcher was one of them. And he was chief of medicine, and I didn't know him, but I thought he would be a good person to speak to.
This was chief of medicine.
My
Where
future.
was he at the time?
At NYU Medical School.
At NYU Medical School. So this is even before your internship you were talking to him?
No,
No. Oh, you went back looking for advice. Okay.
I went back a few months after my internship started. And, it was amazing. I spoke with him, for 20 minutes, and I walked out with fellowship.
That's great.
That's great.
It's like Fred yesterday. He gets a phone call, 10-minute phone call from CDC, and he ends up with a job.
Not just a job, he was a chief.
Chief of some division. Yeah. That's amazing. So
I liked MIT, and it's sort of paradoxical now. I liked MIT because the biology department was so small. It was 3 floors of a building, and everything was mixed: biochemistry, genetics, microbiology. And I really liked that. Nowadays, of course, there are buildings and buildings of biology.
Yeah, you remark in your article that shortly after you got there, that biology basically exploded in the field because of all the molecular biology, nucleic acids translation was starting to get sorted out and faculty increased, right? And some of the people there were Cy Levendahl, Sal Luria, Alex Rich, Darnell, even was Aaron Shatkin there for a while?
No.
He was somewhere else and then left, right? He was here at NIH.
He was in my lab at NIH.
So I wanted to ask you about NIH. So you got this fellowship. How long had NIH been in existence? When was it founded, do you know?
So it evolved. It was originally the Laboratory of Hygiene.
I see.
Which was in D.C. And then after the war, it expanded.
Okay.
And they got the land in Bethesda from a donation to build a medical research facility. So they moved here, and there was a lot of funding after World War II. So I'm not sure the exact date, probably around 47, something like that.
And then they began to support training. I think the idea was to support MDs to do research, right? Initially.
Well, they had a very strong and good clinical research training program.
Yeah, which they still do, right?
They still do, yeah.
But at some point they expanded to support extramural research, right? Which is what Rich and I did for a while, right?
And that was a stroke of genius, because it became, it's still the strongest extramural supporter of basic research.
Yeah.
Yes, so for a long period of time, the NIH was the place to go to if you wanted to do clinical research. Then, of course, when the money started to spread to universities, there was a lot of competition.
Yes.
So
at MIT, who was your research mentor, and what did you do?
Vernon Ingram.
Okay.
And Vernon, for your audience, is known for defining the first molecular disease. And he found an amino acid change in sickle cell hemoglobin. So he was interested in hemoglobins, and when I was looking around for a position, I wanted to do some work with regulation of gene expression. And so Vernon suggested that I look at the switch in hemoglobins from an embryonic to an adult. And there were 2 possibilities, chick embryos or tadpoles and frogs. And I chose the tadpole and frog because if you give thyroid hormone to a tadpole, it rapidly and synchronously metamorphoses into a frog, and it's a free-living creature. So that's what I did. I started out with tadpoles. I would add thyroid hormone to the water, and in a week or so, they would start to turn into frogs. So it was a lot of protein chemistry to start with to define the hemoglobins, because they had not been well defined in tadpoles and frogs. And I was thinking at the time, I guess naively, that the switch in hemoglobin would be like in a phage or a bacteria in which a gene was turned off and a gene was turned on. But it turned out it was nothing like that. The tadpole hemoglobin, tadpole red cells were made in one part of the body, and the adult red cells were made in another part. So it was really a clonal selection type of thing.
Not really naive thinking, though, because in humans and other organisms, it is a transcriptional switch, right? To go from a fetal hemoglobin to a, no?
I think it's different cells, though.
Okay.
It's not just you have a active cell and it turns on and off.
Right.
You know, you're right, it's a turn on and a turn off. All the genes are there.
But associated with a differentiation of the cells.
Yeah.
Okay. So I'm interested that at that early stage, you had this interest in gene expression. Can you pinpoint where that came from?
Oh, well, I think it was 1 year before I went into MIT that mRNA was first described. It was the experiment by Jacob and Mrna and Mrna on rapidly turning over RNA.
Yep.
So it was a very hot idea now to try to do that. And at MIT, Jim Darnell was looking at rapidly turning over nuclear RNA. I actually first asked Jim if I could work in his lab, but he said he was about to move to Sinai, so that didn't go.
Yeah,
but,
you note here that Vernon's style was very different from the one you described earlier, right?
Yeah, so Vernon described it as the British system. I don't know if that was a universal British system, but we sat and discussed the project, and then we didn't talk about it again for a few months.
He'd leave you alone, yeah.
He said his feeling was that that the training for a PhD is training in independent research. And that's how he felt.
But I can imagine that the training that you had prior to that was critical in being able to execute in that environment.
Yes, there were some people that had a little more difficulty in the lab.
Sure.
Yeah,
sure. So you did your thesis on hemoglobin, right at MIT. And then what did you do next?
Well, next I received a letter. I had a deferment from military service. I had, because of the medical degree, I was subject to a doctor's draft.
Right.
So I had a 4-year exemption to do the PhD, but it was only for 4 years. So at the end of that time, I flew down to NIH to see what opportunities I had, because the possibility of joining the public health service would be an alternative service fulfilling the service obligations. So
this was 1966, so Vietnam was just ramping up, right?
Yes, that's correct.
Right.
So you went to NIH specifically because as a PHS member, you could...
Yeah, that's right. I didn't actually go through any of the programs at NIH. because you had to apply like a year ahead of time. So I flew down and I just spoke with a few people, and I happened to accidentally to meet Norman Saltzman. And I say accidentally because I really was on the floor where he was to meet someone else, and I was sitting in the library. The lab had a library, and Norman was very possessive about his library. And I saw this man walk past. I was sitting there, and then he walked past again the other way, and he was suspicious of a stranger in the library. But eventually he came in and he asked me what I'm doing there, and he offered me a job.
That's great.
And you say that hero as well was Aaron Chatkin, Mal Martin, another Salzman, and Mike Bishop.
Yeah, Mike was the one who taught me tissue culture. And I would say it was about a 60-second teaching. We only worked with cells in suspension. And he said, you have a bottle, and what you do each day is you take a ruler and you see how high the level is. And then you make a mark halfway down, and you pour off the top half and put fresh media on. And that was tissue culture. We didn't use hoods. We did everything out on the bench.
Right.
And we used the flame. We flamed everything.
Yeah, sure. Yeah. Actually, when I was a student, we didn't have hoods either. We did it on the...
Sure.
We had a room with UV lights that you would turn on afterwards.
Yeah, I remember at the ICRF doing tissue culture on the bench with the dust bunnies, in the back of the bench and stuff. And as Fred was saying yesterday, it's all about technique.
That's right.
But I never had a contamination. And I thought it was because the HeLa cells could outgrow
the bacteria. Well, you had good technique, because I can tell you that HeLa cells do get contaminated. One of the interesting things, and you were telling us this at dinner, is that Harry Eagle had been there.
Yeah, so this was Harry Eagle's lab. He had left a few years before. However, his staff was still there, the technical staff.
Yeah.
And at that time, they would make all the medium from scratch because of course Harry Eagle defined it with each amino acid in a separate bottle, etc. So they made the medium. I don't think it was commercially available at the time. And for serum, we used horse serum, and there was a farm, and on the bottle of serum, it would say Blackie or someone else, so you could work with the same horse serum.
That's great.
Wow.
That's great.
All the time.
Was Eagle the first to define cell culture medium? Or was yeah. So Eagle's MEM, right? Famous medium. Yeah. We still use it.
Yeah.
So when you joined Norman's lab, it was as a fellow?
It was as a public health research officer.
Okay. And was that that appointment defined as having a certain term to it?
2 years.
2 years. Okay. And then obviously that morphed into a longer-term engagement.
Yeah, that's right. When I went to Norman's lab, I wasn't supposed to work on with viruses. He was purifying chromosomes. I think they were hamster chromosomes because you only have 4 chromosomes, I think. And I thought, well, since I'm interested in regulation of gene expression, and that's where the regulation occurs, it would be a good way to spend a couple of years. But when I got there, he said the chromosomes were too sticky and he wasn't able to separate them well. And why don't I work on vaccinia virus? Because nobody in the lab was working on it. Norman had worked on it, but he switched to polyomavirus. I think everybody, just when I started to work on vaccinia virus, all the people in the field moved to smaller viruses, so that there was hardly anyone left in the field when I started.
This move to smaller viruses, well, there are a couple of things going on. In my time, later on, there was Nixon's war on cancer, and some of these viruses, like polyomavirus, were models for cancer because you could cause tumors and rodents with them. But some of it, I think, was people thinking, well, smaller genome, it's simpler. But no, because they're so host dependent.
Right.
Whereas the pox viruses are quite independent. So in many ways, conceptually, they work out to be similar. I was telling Vincent, I want to tell this story. I was telling Vincent that when I started on pox viruses, which is 10 years later, since 77, I was in Joe Cates' lab, and we had your virus to work with, but I also was interested in getting virus directly from the ATCC so that there was a real lineage that I could count on. But I wanted to understand the lineage of your virus. So I wrote to Norman, and I said, where did this come from? And he sent me back a Xerox page out of his notebook that he'd received virus from ATCC and had his notes on culturing it and that kind of stuff. It was terrific. It's really great.
Why was Norman working on vaccinia virus?
Why?
I mean, what was was it a medical interest or a?
No, there wasn't much of a medical interest because although smallpox had not been eradicated yet, there was no more smallpox in the United States and in many countries. So he was interested in it as a biochemist. Norman was actually a chemist.
Okay.
And so he did some initial work on gene expression. He found some evidence for early and late gene expression. And Bill Yaklik also was working on that. Bill was actually in Harry Eagle's lab a few years before I got there. And
so did you ever work in the same place at the same time as Bill Yaklik?
No.
No. So he had left and he wasn't at Duke at that time, right?
He was at Albert Einstein.
Albert Einstein, okay.
Yeah. I remember when I first started to work in the lab. So Norman actually, he just gave me a vial of vaccinia virus and said I should think of something to do with it.
That's great.
Once again, good training. You had your training in independence, so that's good.
Well, it's also early days that you could do almost anything.
Yeah, when I was at MIT, I learned to do polyacrylamide gel electrophoresis. And at that time, we used tube gels, not slab gels. And a technique that another person at MIT, not me, devised was sort of like a cheese cutter in which you took the cylindrical tube and you made a slice through the center of it.
Right.
And then you could dry that down on filter paper. So if you radioactively labeled the cells and then ran the proteins on the polyacrylamide gel, you can make an autoradiograph, and you'd have a high-resolution print of the different proteins being made. So I had done that at MIT, and the first thing I thought of, I'll do this with cells infected with vaccinia virus and see if I can see a regulation this way rather than previously antibodies were used and enzymes, thymidine kinase, for example.
Got it.
But this could be done radioactively. So that's what I did, and I found that there were 3 stages of gene expression: an early, intermediate, and a late. And it was not until much, much later that I understood what the basis of this was,
Right.
that there are transcription factors, early, intermediate, and late.
You must have figured, however, that this was controlled at the level of transcription, right? And there's also a profound host shutoff, right? So I can imagine that in those, because those experiments are really striking. The regulation of gene expression just jumps out at you. That must have been an exciting result, actually.
Yeah.
So I want to ask you about this virus that Norman Salzman gives you a tube. Where did this come from? Do you have any idea?
Well, he got it from the ATCC, but who gave it to the ATCC? I think it came from the new. Okay, so the strain of vaccinia we use is called Western Reserve, and that originated from the vaccine strain at the New York City Board of Health.
I see.
But in order to grow it better in animals, what was done was to passage it in brains of mice. And it was passaged a number of times. And the WR strain actually is quite virulent in mice. And it grows very well in tissue culture. So it's been the main strain of vaccinia virus.
Okay.
I know from my history with corresponding with Norman that you had passage 63 in mouse brain, right? And that was, I forget who it was. Somehow Joel Dalrymple comes to mind, but I'm not sure that that's true. There was somebody, a researcher somewhere who did these passages and supplied the material to the ATCC. And you get it as it was a lyophilized preparation.
Okay.
That's right. So we continued the circle because I donated then vaccinia virus to ATCC.
Got it. Yeah. So this, is this a derivative of something Jenner used? Ultimately?
Well, the story, you can't really trust any of the stories from that time. But supposedly the New York City Board of Health came directly from the UK, something like within 50 years of Jenner doing that. But whether that's true or not, the early history of the smallpox vaccine is really muddled because obviously they didn't even know what they had. It was just some pustular material. And some people mixed it with smallpox, actually, to try to make a stronger vaccine. And all sorts of things were done.
Yeah, the very earliest history predates germ theory, right? So they had no idea what they were doing.
So today, where do you find related viruses? In rodents, you said last night, right?
Yeah, most of the orthopox viruses, which is the genus which vaccinia and smallpox viruses, mostly they're rodent viruses. Okay. But there are other pox viruses that infect birds, insects.
Right.
Just to make sure that the audience is up to speed, we've done this. Matter of fact, my first appearance was TWiV 26.
Wow.
Okay, 560 episodes ago. but so we've done pox viruses a number of times. But the important thing here is that vaccinia that we're talking about is the virus that was used as the vaccine for smallpox. So it's essentially an attenuated, in a way, an attenuated smallpox. It's a related pox virus, so you can get cross-reactivity without getting serious disease. But as we've already discussed, the actual origins are murky.
Yeah.
That's right. People think now probably it's a virus close to horsepox virus.
Yeah. And so there's a fellow in Brazil who loves to research the origins of the vaccine. He got the vials from eBay.
Yeah, and I'm forgetting the name, but we did a whole episode on the history of the vaccine.
There was a sequence of a very early vial of smallpox.
Yeah, right.
And it's close to a virus which was isolated once from horses in Asia.
Right.
And
It's not exactly the same, and it's not a descendant, but probably it's close to it.
And your friends, you both know Clarissa Damaso, right? She's also interested in this. She works with him and writes about it frequently. When you began working, did we know at that time that this virus replicated in the cytoplasm?
Oh, yes.
We did. What was the evidence for that?
Oh, well, vaccinia virus was the first virus that was observed by electron microscopy. And so you could see the particles in the cytoplasm.
I see.
Although for a number of years there were questions whether some of the replication is in the nucleus. And probably through the early 70s, it was still a question.
Okay.
Dick Moyer did some experiments to show that it's not at all in the nucleus.
Right. All right. So then you had a 2-year appointment, but I guess they extended it, right?
Yeah, they extended it. Yeah, so when I came, I thought working with vaccinia virus was okay for 2 years. But I got more and more absorbed into it. And especially, it was in '67, in the spring, there was the first Animal Cells and Virus Gordon Conference. And I didn't realize at the time that this was the first one. I thought, it had been going on for a long time. But Bill Yaklik convened it. And it's interesting that he's also the convener of the first ASV meeting. So anyway, at that meeting, Joe Cates and Bill Munyon gave short talks that vaccinia virus had a transcriptase in the virus particle. And that really excited me and presumably everybody else, because here I thought I could use my biochemistry training, interest in gene regulation, and try to isolate and characterize all the enzymes needed for transcription, because they're all packaged in one place in a virus particle. I wouldn't have to go to a cell and have all the mixture of cellular enzymes. I could just do it all from the virus particle.
Just by purifying virus, you got a several thousand-fold purification of all those enzymes.
That's right.
And was this the first transcriptase in any virus that predates VSV and all that? Okay.
Yeah, so the story went, so they did it first in vaccinia virus, and then the next year, Aaron Shatkin showed reovirus had a transcriptase.
Okay.
And it's a nice story how he did it. Anyway, well, just to say, he incubated reovirus with nucleoside triphosphates, and then I think he did TCA precipitation, and he put it on a counter, and it was getting late, and he came back the next morning, and he saw this burst of counts. So that was really fun. And I was in the lab at that time also, so I was looking over his shoulder.
Does it at NIH this happened?
He was at NIH, yeah.
Wow.
Yeah, and then afterwards, the VSV came next, and then the retroviruses.
Okay. Let me just put in here, because I looked it up. It's episode TWiV 478, A Pox on Your Horse. Okay, when we talked about synthetic horsepox and the whole history of the vaccine. Right. And the guy you were mentioning is Jose Esparza. Right. Okay, who does all of the archival vaccinia.
And he had found a vial of an old smallpox vaccine on eBay, right. And he sequenced something from it, right? So this meeting basically got you more excited in vaccine.
That got me more excited. So,
That's interesting.
so what I decided to do, so viruses, of course, are sort of like nuts. They're built to withstand the environment. So you have to get the enzymes out. So, so I started to, in the lab, try a variety of different detergents and urea and all sorts of things. And if you just use non-ionic detergent, they don't come out. If you use SDS, they come out, but they're inactive. And then through trial and error, I found that deoxycholate, a certain concentration of sodium chloride, and dithiothriotol, all the enzymes came out, and the structural proteins were still insoluble. So I just had to spin it out, and the DNA and all the enzymes were there for me. And then I discovered that if I just took a small DEAE cellulose column with 0.25 molar sodium chloride, the DNA would stick and all the enzymes would come through. So then I had what I needed, and the fun thing then was to make assays to try to find out what enzymes are there.
Right.
And so one assay was ATPase. Is there an enzyme that can cleave ATP? And so we found, yes, there was, but there wasn't activity after I removed the DNA. So we looked for a DNA-dependent ATPase, and we found it, and then an RNA-dependent. And that was the time Enzo Paoletti came to my lab. And so we would work, we would really work in tandem. We didn't know how stable these were, and there was such little quantities that we'd never stop a purification. One person would be in the daytime, the other would come in the evening, and we never froze anything or did it. So it was amazing. It's a tiny amount of protein, and we purified all these enzymes to homogeneity.
So how many enzymes today do we know are in the particle?
Oh, I don't know the number. More than a dozen.
Yeah.
And how many total different proteins including structural proteins?
80.
80 in the particle. And you call this a bag of enzymes?
Well, yeah. Well, after I solubilized it.
Yeah. But that was your name, a bag of enzymes, right?
Probably.
So I want to talk about the capping enzyme, but there's a story before that, right? Which is about mRNA capping itself. Okay, so tell us the mRNA capping story.
Okay, so I think the story comes from two directions. So it was known for a long time ribosomal RNA was methylated, but it was not known that mRNA was methylated. And the development of oligo-DT, cellulose and poly-U, allowed, well, I should take one step back. Mary Edmonds showed that there's a poly-A tract at the 3' end of cellular mRNAs. And then after she found that, other people showed that you could purify mRNA because of the poly-A using oligo-DT or poly-U. So now for the first time, cellular mRNA could be obtained in sufficient purity to see whether it's methylated. And in 1974, Perry and Kelly was his technician, published a paper saying there's about 2 methyl groups in each mRNA molecule. And since at that time we already knew that vaccinia mRNA had a poly A tail, and we knew that vaccinia mRNA was translated in the cell, it seemed obvious that I should look to see whether vaccinia mRNA is methylated. And just at that time, Aaron Chatkin, who was then at the Roche Institute, was at NIH visiting, and we were sitting talking, and he told me that a scientist in Japan, Mrna, had found that cytoplasmic polyhedrosis virus, double-stranded RNA in the particle, had some kind of a structure at the 5' end, so they couldn't sequence it.
Okay.
So Aaron mentioned that, and he said that a student of Mrna's was going to come to his lab, and they were going to look at reovirus. And I don't know whether Aaron was going to look at mRNA or the double-stranded RNA, which is what Mrna did, but I told him, well, did you see Bob Perry's paper? It's in the first issue of Cell, by the way.
First issue, wow.
And I said, I'm planning to see if vaccinia mRNA is methylated. So before Aaron left, I said, well, I'm going to put radioactive S-adenosylmethionine into the transcription system. And Aaron said he was going to do the same with reovirus. And then a week later, I called Aaron up to say, vaccinia mRNA is methylated. I had to leave a message on his answering machine, but a couple hours later, I got the return, reovirus is also.
Wow. But you didn't know where the methyl was, right?
At the first step, I didn't know, but I knew there was 1 methyl per 1,000 nucleotides. And that's the average size of vaccinia mRNA. So,
and you knew that the 5' end of some other messenger-like RNA was blocked. So
no, I didn't know that.
Oh, okay. I thought the Japanese group.
No, that wasn't the mRNA.
Okay.
It was
the
double-stranded RNA. But yes, you could extrapolate and say that it's probably the 5' end. But
so how did you get from the methyl to the cap then?
Okay, so we had a huge advantage. So, for example, over cell biologists, because we had pretty pure systems in which we were making mRNA, and we could add radioactively labeled nucleoside triphosphates. And at that time, it was available to get for example, nucleoside triphosphates with the alpha p32 labeled, or the beta, or the gamma.
Right.
So when we put that in, we could determine that the beta and gamma p32s were incorporated into the RNA. And okay, and we also had the advantage of, which Aaron didn't have, of a soluble system. So we very quickly showed that we could add exogenous RNA and have it capped.
So are you talking about an infected cell extract or the virus part?
Virus.
So you can crack them open and make mRNAs?
No.
Yeah.
You could you could make mRNA with a virus, but you're talking about solubilizing enzymes and getting.
We did that almost immediately.
Okay.
But we initially used almost the same system Cates and Munyon had used, which is just the core of the virus.
Right. Okay.
Yeah. So then it became a question of what's the structure of it, and that was interesting because, I was not a nucleotide chemist. Well, actually, I did. When I worked at NYU, I published a paper. Maybe that helped.
Yeah, sure.
So anyway, it was quite a challenge to figure out, to run columns and figure out. But what really helped was this P32 labeling, that we knew exactly which phosphate was going into it. So Aaron and I were working on it separately, and so at some point, so in '74, the same year Bob Perry first published there was methylation mRNA, Aaron and I both published the same year that the viral mRNAs are methylated, and the next year we both had the structure of the cap, and it was sort of fun because I said, I'm pretty sure I have the correct structure, and he said with Rio he had, and we didn't tell each other, but we both submitted it. Dave Baltimore actually put my paper into PNAS. And I forget who did Aaron's, but at the same time we sent it in, we sent each other manuscripts, and the structures were almost identical. Reovirus had the M7G triphosphate G, and Vaccinia had G or A.
I see. But the M7 was up at the
M7 triphosphate was the same. So once that information was there was just a deluge. My own lab collaborated to show herpes virus, adenovirus, histone mRNAs are all capped. And Aaron's lab did a lot of work on the role of the cap for translation. Whereas my lab, because we had a soluble system, we purified the enzymes and determined what the mechanism of capping.
And once again, that capping enzyme is in the virion. It's part of the soluble extract of the virion.
So I was just talking with Bert Flanagan, and a couple of years later, they're looking at polio for a cap, and there's some weird structure that turns out to be a protein.
Yeah. So I tried polio also, and I couldn't get any label into the 5' end, so I never published it. I should have published it as a negative.
Yeah. So you never got any gamma or beta label into the RNA, right? Yeah, that would make sense. Oh, that's very cool. So obviously all your biochemistry training is helping you a lot, right, in this work, this bag of enzymes. Now, so we said before, you can take the virus particles and they will make mRNA under the right conditions, right? Yeah. They make capped and polyadenylated mRNA. And now, so I'm going to ask this question now, if it's okay with you. So is that why if you take the DNA and put it in cells, it will not initiate an infection because the promoters aren't recognized, or? Yeah,
exactly. It has its own transcription system. The promoters are not recognized by
the
cellular polymerase system.
That's, I mean, on an evolutionary basis, that's fascinating how that came about, right? How it diverged from the cell. So
I think this afternoon you could ask
Eugene
Kuhn.
Yes. Maybe they were there first. So none of the viral promoters are recognized by cellular polymerase, right?
Right.
And so if you put DNA in, you have to have. So what's the minimum you need for DNA to start a cycle? What do you need? One transcription protein or more than one?
No, I think there's 18 polypeptides involved in transcription. There's 8 for the RNA polymerase, transcription factors. I think it's 18, if I remember
Something like that,
right.
Yeah.
So I think now when you want to rescue a DNA, you have another pox infection, like an avian pox
That's
virus, right?
Yeah.
In the horsepox rescue, they had to put that in a avian pox virus infected cell. Otherwise, okay.
To get the whole transcription thing to work right, you really need to have it in the context of an intact core. Okay, there's some structural components there, and that's not entirely true. I mean, you could later on,
transcription.
For early transcription, you can get it done in a soluble system.
Is there any nuclear function required? If you take out the nuclei from a cell, can you infect it with vaccinia?
Yeah, so there's a couple of papers on that early, and the papers said enucleated cells can replicate the genome of vaccinia, but you don't get infectious virus out.
Right.
And no one really, these were very early papers in the 70s, and it was not looked at what the block was or whether the DNA was intact or complete or what. So there is still a question of whether any host proteins are involved in DNA replication. And if there are, I don't think it's many.
Mm-hmm.
So, Rich, what do you want to talk about next? That was a good story.
There's a phase of your career, if I understand it correctly, that sort of proceeds from the bag of enzymes phase of the career that's gene expression, right? Where there's a lot of, now you're really getting down to the nitty gritty of transcriptional control of gene expression. And so, what were some of the highlights of that phase?
Okay, well, I think the next stage actually was cloning.
Okay.
And of course, you will know that Paul Berg cloned SV40 DNA, but then everybody became alarmed at the possibility of E. coli then becoming dangerous. And so there was a moratorium on cloning. And then the NIH put out guidelines. And the first guidelines were very strict. To clone anything from a virus, you had to use a BSL-4. So essentially, there was no research being done. And then the Recombinant DNA Committee of NIH came out with new guidelines that viral DNA, as long as it's not a whole intact one, could be done at BSL-2. So that made it possible. So just before that time, Ricardo Wittek, who was doing poxvirology in Switzerland, in Zurich, he was a young person, and he came to my lab, and we decided we were going to clone vaccinia. So it's interesting. At that time, you had to clone it in lambda. You couldn't do a plasmid because the lambda vector was considered safe. And so what Rico and I did is we cut up vaccinia DNA with restriction enzymes. And actually, that's another story. Restriction enzymes just came around also at about that time. We cut up the DNA fragments, and we had the lambda DNA in 2 separate tubes. And then on the day that it was legal to clone, we put the two together, and we had our first recombinant DNA on the first day it was possible to do it.
January 2nd, 1979.
Yeah.
I know that's my birthday, January 2nd. And in that year, I was going to David Baltimore's lab, and he said, now you can clone the whole polio genome. So let's do that, because you couldn't before January 2nd.
Yeah. Yeah. So
you had cloned the entire genome in fragments, right?
We didn't do the whole genome on January 2nd.
Yeah.
That took a while longer. But we did, and also, I mean, everything was coming together, restriction enzyme, cloning, sequencing.
Right.
Not the Sanger sequencing, the Maxim Gilbert sequencing. So all that came together at the same time, and we were able to start to look at what is a vaccinia gene.
Right.
And what about the ends of the DNA? Because it had been known that you can't separate the two strands of vaccinia DNA, and we were able to sequence around the hairpins and define what the structure of the gene is. And I mean, I should say I had great postdocs who were doing that work.
Yeah.
Well, when you mentioned Rico, I can see in my head, I can see his gels, because technically he was so skilled.
Yeah, he had the best gels. He made his own apparatus. And the reason it was so great was that it didn't have any rubber seals or anything. It was all one piece made of glass. It was all fused together.
Wow.
And, you know, every once in a while it would crack or something and take a new one. But they were made in the shop here at NIH.
Those days when we had shops. You still have a shop here?
I don't know.
Yeah, we didn't.
I think the shops, if you really wanted to make something that was, you know, not commercially available.
So then you spent a long time really dissecting the promoters and the enzymology of the transcription, both early and late, right?
Yeah, right. So one of the first, I had a great postdoc, Andrew Davison, who's still working now in Scotland. And he was the most organized person I ever had in my lab. And at that time, you have to realize it was in the 70s. He defined the vaccinia promoter by making 350 recombinant viruses in which each nucleotide was exchanged for each of the other ones.
Wow.
And we had a reporter system, beta-galactosidase. So he had these 300 and he determined the level of expression. And from that, we defined the promoters.
That's just an incredible amount of work.
Tell me how it works. You said originally there are 3 waves of gene expression, right? So I presume the first is based on the transcription factors in the particle, right?
Right. Okay. Then the next is the dependent on replication of the genome.
Okay. The middle wave.
That's right. So among the early proteins, so there's about 100 early proteins that the virus makes. And among them is the RNA polymerase, capping all the enzymes needed for transcription. But the early proteins have the transcription factors for the second wave. Right. So when the DNA is replicated, you then start to make intermediate gene expression. And then some of the proteins of the intermediate wave are transcription factors for the late phase. So you have this consecutive early, intermediate, and late. And then among the late proteins are the early transcription factors. So they get packaged into the virus particle so you can start over again.
Got it.
So it's kind of confusing a little that the early transcription factors are late proteins and the intermediate are early proteins.
Right. So among those middle proteins are the DNA polymerase, I assume, right?
The early.
The early includes a DNA polymerase.
Yeah, because you have to make DNA
before you get middle.
Before you can actually express it.
Got it.
However, in the absence of viral DNA replication, you can transfect in a plasmid with a promoter, an intermediate promoter, and it will get expressed because the early proteins have made the protein, and it's just
Right.
waiting for
Right.
a template.
Right. How many genes are involved in DNA replication? A lot?
So there are 7 that have been isolated from vaccinia virus directly, not counting enzymes for precursor
Yeah,
metabolism.
So I guess it has to make all its own triphosphates, right? Because is there a pool in the cytosol that it draws
Yeah,
from?
there's a pool
There's
of
a pool.
cytoplasm.
And there's just one DNA polymerase?
There's one DNA
So
polymerase.
we don't need a primase or...
There is a primase.
There's a primase and a DNA polymerase, okay. And the polymerase is a
There's
primer?
a primase helicase.
Okay.
Yeah.
So
So Eugene actually is the one who helped us to find that. He discovered by looking at sequences
Yeah.
that one of the vaccinia proteins had a very slight degree of homology. If you just did a blast, you'd never pick it up. But he picked it up by doing reiterative searches.
It was known for a long time that gene was involved in DNA
It was
replication.
a helicase.
Right,
And
but
it was
we
an
didn't.
ATPase and presumed to be a helicase.
Right.
But it wasn't known that it was
Right.
a primase.
Right. So that was a real breakthrough to figure out, oh, it's a primase.
Yeah.
It's great.
Yeah, so he told us, and then we showed that it was a primase. We synthesized it in vitro, showed it was a primase.
That's what Eugene is good at, looking very closely and for a long time. And yeah, there's a little hint of this, right? It's amazing. Oh, so so Rich, you have here entry proteins.
Well, I want to, before we get there, I think it might be appropriate to talk about recombinant viruses.
Okay.
Because when did that start up and what is it?
So again, there's, just like with capping, there are 2 stories, and a story from my lab and a story from Enzo's lab. So in my lab, it was natural that we had defined where a promoter is and what a promoter, basically how long it is, etc. So it was pretty obvious that we could take a coding sequence of some other gene and we'd be able to transcribe it if we put it next to a promoter. And so we did that, and the other idea was also that because vaccinia virus was a smallpox vaccine, if we put in a gene from another pathogen, we would have a vaccine.
Right.
So we entered it from the transcription starting point. And Enzo, and also from the cloning, it was right at the same time we did the cloning. And Enzo started from the point of view that I think right from the beginning, he wanted to make a vaccine.
Mm-hmm.
And he shotgunned foreign DNA into vaccinia virus without cloning or anything. And in order to get expression, he put a herpes virus promoter before the gene. And he got expression. It turned out it wasn't because of the herpes virus promoter. It just went into a fortuitous place in the genome of vaccinia virus where there was an upstream promoter.
Right.
But anyway, both our labs then started to show that recombinant vaccinia virus could provide protection against infection of other pathogens. And probably the highlight of our work was putting in the hepatitis B antigen into vaccinia virus. And we showed that chimpanzees could be protected against HBV infection.
Now, my understanding now is that this whole technology has evolved, and there are licensed veterinary vaccines that are recombinant poxviruses.
That's right.
Rabies, and I don't know about others.
There's a lot of them. There's about a dozen veterinary vaccines.
And there have been...
And there's 100. It's amazing. One of my postdocs looked it up on clinical trials, and there's 114 clinical trials with poxvirus-based vaccines.
Yeah.
But none licensed in humans.
None licensed.
And do you, hard for me to believe that that's because they failed trials. What's the barrier?
The HIV ones did. They don't give any protection.
That's a tough.
Well, there was a little bit.
Yeah, a little bit. Yes, very little. Yeah, that was
but none of the other vaccine strategies have worked either for HIV.
Right. That's a tough.
That's an HIV problem. But one of the coronavirus, one of the new vaccines is an MVA-based vaccine.
Yeah, there's a whole lot. I mean, now people are doing every possible one.
Yeah.
The one the NIH is doing, I think, is the RNA lipid one.
Recombinant poxviruses have been good research tools, however, in addition, right?
Yeah, for I mean, that was the main thing. After we we actually made a toolkit for people to put genes into vaccinia virus. We sent out, I think we sent 1,000 of them out. Plasmid with a restriction site for putting in your gene, and it would go into the TK region, which would be selective.
Right.
So for immunology was the main thing, because that was the first time you could look at T cell epitopes. Right. Because you have to express them not as a protein, but intracellularly. So, so that was used for mapping T cell epitopes. So even if it never makes it into a licensed human vaccine, there was a lot of work which, in fact, pre-vaccine helped.
Yeah, I think it will at some point. It's just a matter of time. We have plenty of viruses to go on.
Right.
Yeah, but people now, there was a time where vaccinia was unique. Now every virus is a vector.
Of course, yes. Many are being developed as vectors. But this MVA, modified vaccinia Ankara, is interesting because it was selected to grow in chicken cells, right? It doesn't do, and your most recent paper is figuring out how that works, that host restriction, right?
Yeah, that's right.
So you continue to work on that.
Yeah. So there are 2 papers we just published which show, so MVA lost about 15% of its genome during several hundred passages, and it had, in addition to deletions, it had mutations in 114 other genes. So the question was, why can't it replicate in human cells? And we showed that if we put back 2 genes, it can now replicate in human cells.
Wow. I think I was talking with Rich about that. I think that's great.
And we're still working on the host. We have one of the host factors which is restricting it.
Do you mind if I ask about T7 at this point?
Go for it.
We're on expression.
Yeah.
So at some point you developed this T7 system. Yeah. And so why was that done? Is it better than vaccinia alone?
Well, I think it was Ed Niles, actually, first idea to put T7 RNA polymerase into vaccinia.
Right, and I thought he was nuts. I thought that was just a crazy idea.
Yeah, and he was I don't know whether he was trying or not, but we had a conversation, and I said, well, we could do it easily. And he said, okay, we'll make it a collaboration. So it was three-way between Studier, who cloned the T7 RNA polymerase gene, Ed, because it was his idea, and our lab, because we had the know-how to do it. So, so the first thing was very simple, and it was just to put T7 RNA polymerase into vaccinia, transfect in a plasmid with a gene under a T7 promoter, and you'd get expression. And instead of being a nuclear system in which the RNA has to be processed and everything, this would be expressed in the cytoplasm.
Got it.
So it works 100% of the time. So that was the stage with Ed. Then we decided to, well, let's make a recombinant that also has the gene under a T7 promoter. And we couldn't do it because the expression was so high that the vaccinia, it inhibited vaccinia replication even if it wasn't a toxic gene. It was just so much made. So that then led us to regulate the gene, and we used the E. coli lac operator system. I think it's the first use of it in a eukaryotic type system.
I see.
And so we put the lac operator next to the T7 polymerase and the lac operator next to the gene. So we had a double way.
Got it.
And we put the repressor into vaccinia virus. So we had this whole eclectic system, and it worked. And in fact, so originally we used it for overexpression.
Right.
But then we started to use it because the regulation was so stringent when we didn't add IPTG that we could use it to make conditional mutants. So if instead of putting in a foreign gene, we put the operator, the lac operator next to a vaccinia gene, if we didn't add IPTG, we wouldn't get detectable expression. So we had a null mutant. And so that was very powerful. Of course, as you know, Rich supplied the whole poxvirus community with TS mutants. And the TS mutants were very valuable, but with the inducible system, you could do it more systematically.
You could really target a gene.
Yeah.
You know, and that's what's missing from the classical genetic approach.
Now, in these vectors, you have an internal ribosome entry site, IRES. Was that present from the start or came in later? And if so, how did that come about?
Okay, we did that because of our knowledge of capping enzyme.
Right.
We knew that the capping enzyme is associated with the vaccinia RNA polymerase, and it's co-transcriptionally capped.
Right.
So that we thought if we're going to express from another RNA polymerase that doesn't have capping, we have to eliminate it. In fact, we had done experiments without the iris, and we did get pretty good expression. But when we looked at the amount of mRNA we had, it was just a huge amount of mRNA, and a small amount was getting capped, probably by soluble capping enzyme.
Okay.
But then when we put the iris in, it boosted it way up.
So obviously the iris had already been discovered by that
Yeah.
point, and you just thought this would be a way around the cap.
Yeah, it hadn't been discovered very long, actually.
Yeah.
We got it from Ann Palmenberg.
I see.
She sent us the piece of DNA.
Yeah.
So, yeah, again, all these things were,
That's how science
all
works,
happening.
Right?
And there was a period of time when the recombinant vaccinia, in particular vaccinia T7, was used by a large number of laboratories to study expression and processing of various different proteins. I in particular remember, was it flu hemagglutinin or one of the trimer surface proteins that was, I think it was flu, that was expressed using a Vaccinia T7 system so that you could look at the pathway for processing and presentation of that protein.
So we had a project a number of years ago where we wanted to overproduce a rhinovirus protein that altered membrane structures in the cell, made these vesicles. So we were doing the Vaccinia T7 and then electron microscopy. And my student comes in one day with the EMs and she said, what are these big things in the cytoplasm? Well, that's the Vaccinia virus.
Actually, I was surprised that some of those experiments worked because Vaccinia is, it's a wrecking ball in the cell,
Yeah. Yeah. So Vincent, you'll be interested. We never published it, but we put polio. It was very shortly after you did the polio work. We put the whole polio from a plasmid with a T7 promoter and transfected it into cells with Vaccinia.
And that worked,
polymerase. Right? Well, sort of, except the polio killed Vaccinia virus.
That's right. It would expect it to. Yeah. So we had put DNA in, as you know. We put a plasmid in. And then a year later, Eckhard put in vitro, he did T7 transcription in vitro to make polio RNA, and that was more efficient because RNA is better.
Yeah, I think that's when we did, after Eckhart did that in vitro, we said we can do it more efficiently.
Yeah, no, that would be a perfect, I might have heard about that or.
Well, we never published it and I don't think I ever spoke about it.
No, but that makes perfect sense, to do it that way. Yeah, as long as you don't work, because
we would have had to knock out the, what's the factor that inhibits translation?
Yeah, 2A.
Yeah, I think if we knocked that out, we could have made it.
Yeah. I mean, I think we were fooling with that too at some point and never got anywhere. But that's an interesting way to do it, is be very efficient. Yeah. Okay. What do you want to do next?
I want to talk about the origin of the viral membrane, because this is a long-standing mystery in poxvirology that in the last several years you've made real progress on.
Yeah. So Sam Dales took the first well, he didn't take the first, but he took very good electron micrographs of Vaccinia-infected cells. And the first thing he saw...
Now, this is back in the 60s,
This is in the 60s, the early 60s.
Yeah.
So what he saw were membrane crescents. And in the cytoplasm, and they weren't attached to anything. It looked like the ends of these crescent membranes were free. And he interpreted it as a single bilayer with spicules on the top. And nothing happened with that. I mean, that's how it was for the next 20 years, I think.
And he called it de novo membrane biogenesis.
His interpretation, which based on the experimental system, he said that this is de novo membrane synthesis because it's not attached to anything. Okay, and then I don't know how many, in the 90s, so it was even longer than that, some cell biologists who started to work on Vaccinia virus said that's impossible interpretation. That, first of all membranes come from pre-existing membranes in the modern world, and that there's no such thing as a bilayer with free ends. It's unstable, can't be. So they started to show electron micrographs, which they interpreted as double membranes, but very tightly opposed. And only their resolution was enough to see that. Well, at the Pox meeting then, Sam Dales was still active at the time. There were these fierce arguments between the two groups. And essentially, it was all pox virologists on Sam Dale's side and this group of nouveau pox virologists on the other side. And there was a lot of data from my lab and others in which you could very clearly see it was a single bilayer.
Right.
And so that went on.
That went on for years.
It went on years with review articles being written by that group on, how the membranes really are and all that. And it got into textbooks that it's a double membrane. But the pox virologists, I don't think, ever believed it. And finally, the group did tomography, and they said, what do you know? It's a single membrane. And they published it in a high-profile journal, as if, they said that there had been a debate for many years of whether it's single or double, and now we show it's single. And they were the only group that was saying it was double. I think you got annoyed at that.
Oh yeah, sure, yeah. There was a problem with that. Yeah. So, but you still have the problem.
Okay, so you still have the problem. So now they are single membranes. So how do they exist? Well, the idea that they're free ends, well, that's easy. There's protein or something can be covering it. But where do they come from? So, so we were making a lot of mutants. So this was sort of in my lab, it became a project for graduate students. New graduate student, new gene, use the inducible system and find out what that gene is doing.
And this is a standard sort of genetic microbiological strategy. Knock a gene out, or genetic strategy. It doesn't have to be microbiology. Knock a gene out, ask what's wrong, okay, and then from that, reason backwards to what it actually does.
Okay, so we also had a very good electron microscopist. We still have Andy Weisberg, and she's been working in my lab for, I don't know, more than 20 years, and she's very good at recognizing the EM structures. So with one of the inducible mutants, she saw membranes that looked like they were connected to ER. And then we did more experiments, and we showed what was happening actually when that gene was not expressed. The viral membrane was being made in connection with ER, and it was actually butting into the lumen of the ER. So we had these particles in the ER lumen, and particles incomplete were still connected. And then we found several other genes that are also required for the assembly. So we now have a model in which the ER is cut, and these proteins, they're all very small proteins, and we can see they're attached at the ends. Actually, you can't talk about ends. It's a sphere, so it's sort of
so it's a rim of a cup.
A rim. Yeah. So this protein is attached to the rim of it. And so what we don't know now is how that ER normally gets broken into pieces, because we could only see the connection because it failed to break. So there are 5 proteins we found that are required for this, but none of them have a motif like a GTPase or an ATPase. So it's unlikely they could actually be cutting. You need energy to do that. So it must be in concert with a cell protein. And what that cell protein is, we don't know.
It's not any of the escorts.
We don't know what protein it is.
Are these late viral proteins or?
They're all late
Oh,
viral
late.
proteins.
So, related to this, there's, for me, a very amusing thread in your work that goes all the way back to the beginning with rifampicin,
Yeah.
okay, and winds up with the spike protein. Could you? I'm interested to hear your take on that trail, because it's very interesting.
Okay, so rifampicin experiments were the first interesting experiments my lab ever did. And there was a paper in Nature, it must have been 1967 or 68, something like that, just shortly after I arrived at NIH, in which they showed rifampicin inhibits vaccinia virus. And rifampicin is a specific inhibitor of E. coli RNA polymerase, but not eukaryotic RNA polymerase. And the paper purported to show that it was inhibiting vaccinia mRNA. And I thought, that's great. If I have a specific inhibitor I can use, be terrific. So we started to work on it, and we didn't get any inhibition of vaccinia mRNA. And it turned out, this is also a lab that was not a poxvirologist lab that published in Nature. And they weren't getting one-step infections. My interpretation is they saw a decrease in mRNA because they were blocking the spread of the virus. And it turned out that rifampicin is a very, very specific inhibitor of the poxvirus membrane assembly. And specifically, it prevents putting on what Sam Dales calls spicules, but which is actually a capsid-like protein. It forms an icosahedral layer around the immature virus particle. And it specifically prevents the interaction of that protein. And also just a year ago now, maybe 2 years now, the EM and other physical studies have shown that rifampicin interacts with that protein that we had shown formed the icosahedral capsid. We knew it interacted because we made point mutations in the protein, but we didn't actually demonstrate the interaction.
What I love about this story is that it's this path that goes throughout, the whole 50-year career. It starts out with maybe...
You have to be working on the project for 50 years to go back to
That's
it.
right. It's amazing because it starts out as, well, maybe it's a transcriptional inhibitor, but no, now it inhibits morphogenesis. Oh, it targets this gene D13. We don't know what that is. And then you figure out that it's the spicule protein, and now it's really important in the morphogenesis. And this some sort of weird accident that rifampicin, which is otherwise an inhibitor of transcription. Is that a fungal protein of some sort?
I don't know of any poxviruses that infect fungi that it would have naturally made an inhibitor.
So it must be a real accident that it actually targets this.
It's an antimicrobial, right? It's used as an antibiotic, right?
Yeah, but it's used for TB.
TB, yeah.
But the affinity for the D3 protein is not high enough. It's very reversible. You wash it out, and in seconds, it starts to go back. So it was clear from the beginning there was no use for an anti-poxviral drug.
So a somewhat related topic is the entry proteins. That's been another major thing of the
Yeah.
past decade or so.
Yeah, so that started out the same way. Tanya Sankovich, rather than a student, was interested in the conserved proteins that look like their membrane proteins. So she made an inducible mutant of one of them, and she expected to see a defect in assembly of the virus. Of course, that's what we found with other membrane proteins.
And there's like 20 or so membrane proteins, right? For
40 predicted.
40.
My God. More than herpes.
But she honed in on those that were conserved in all poxviruses.
Okay.
Okay. So the first one she did, it was required. If you don't express it, there was no infectious virus. And we did EM, and we thought we would see a block, but there was no block. So then we showed that the virus particles were normal looking, and we could purify them, but they were non-infectious. And then we showed there was a block in entry. Okay, so then...
So those particles would actually bind to cells, right? Is that correct?
Let's talk about that later.
Okay.
That's a question.
Okay.
Yeah, so that was the first one. So then she tried another gene that also was conserved, exactly the same phenotype. And then a postdoc came in the lab, and I told him, why don't you try these 3 genes? And they all had the same phenotype. And then at that point, we began to suspect that it was a complex. So what Tanya did was to use an affinity tag, and she pulled out all the proteins associated. Okay. And to make a long story short, there are 11 proteins that together form a complex, and each one of those is individually required for entry, and they're required for fusion of the viral membrane and the cell membrane.
Do they all have transmembrane domains?
They all have transmembrane domains. And we don't know the full structure. 2 of them, there are crystal structures, and we know some binary interactions, but nothing looks like a type 1, 2, or 3 fusion protein.
Okay.
So we assume that it's this complex, not individual proteins that are somehow putting it together.
Okay. Does this fusion happen inside the cell or at the plasma? Both?
Yeah.
Okay.
Yeah. So
do we know if there's a specific attachment receptor?
No. So it is known that there are a few proteins that bind to glycosaminoglycans. So that, attaches. And there's one paper suggesting that one of the entry proteins, L1, if you don't have glycosaminoglycans, that helps the association.
Okay.
But what any of these proteins are doing individually, we don't know.
Are you still working on that?
We're still working on that, but our main thing now is we're going to try to get a structure of it, because it's easier to get a structure without a structure, it's hard to know what to do.
You can use cryo-EM?
We haven't yet. I've been thinking about it. Before that, we're going to be we're doing a cross-linking project to define. Paul Gershon did a little bit of it, but we want to see all the interactions And you can do that now with high enough density that you can actually make a three-dimensional model. It was done for the proteasome, for example.
Can you get sufficient resolution by microscopy of some sort to see a difference between virions that do or don't have the fusion complex?
Yes. So you weren't at the poxvirus meeting last. So Mercer's lab in the UK used the high-resolution fluorescence microscopy, and he provided evidence that these fusion proteins are at the tips of the virus. So the virus isn't a sphere; it's more a cigar-shaped sort of, and they're at the two ends. And he used one of our mutants that doesn't express one of them, and now they're no longer localized at the end. They're still in the membrane, but they're diffused in the membrane.
Jason, what is this?
Jason Mercer.
Mercer, right.
Yeah, he published that.
And we actually did that paper on TWiV.
Yeah, we did, yeah. And
I was looking for resolution enough to see little blobs.
No, you don't see anything. So I don't. Yeah. So what we want to do, but we haven't even determined the feasibility yet. But we're planning to make collaboration to see if the affinity purified complexes we have a structure that can be determined. The cryo-EM now is so powerful, you don't have to have a homogeneous structure either.
That's right, yeah.
TWiV 507, fusion of form and functions. Where
not too far, not too long ago. I remember that. And then he wrote in afterwards to clarify some things. Yeah. Should we go to a few more general questions? Or you have anything else?
Actually.
Yeah, go ahead.
When my lab was just winding down, we had collected a bunch of data to do, because we got very interested in the internal core structure, which is neglected in for a long part of Vaccinia's history, because it takes care to preserve that structure for microscopy. And we were closing in on a 3D tomographic structure of the virus that included the core, but never finished it. Have you considered that or made any progress in that realm?
No.
Please do it. Because I want to see that.
No, I think the things, so what are the major questions you may ask me that I would like to, get some more insights. And so one of them is we talked about assembly. What is it that cuts up the ER membrane? That would really be interesting. And the other is the entry complex. And so I think that occupies my time.
Right.
I won't be able to do your favorite experiment.
Sorry.
Okay.
Sorry.
Well, maybe we can get somebody else.
There's also a lot. I think we have the outlines of most of how the poxviruses assemble and replicate, but of course, in each one, the details are not there yet.
So you've been here for 50 years at NIH. Obviously that's been good for your career. Did you ever think of going to an academic institution?
Yeah, so I've had a few opportunities, and I would, take a look at what I have here and what I'll be getting there. And, even a Howard Hughes position I was once offered. When I put it on paper, at that time I had a very large lab here. Now it's much smaller, but it would have been difficult. Even with a Hughes grant, I would have needed a couple of NIH grants also to do that, and everything was fine. For me, I wouldn't leave unless there was both a pull and a push.
Right.
And NID has been so good to me that I never had a push.
Yeah, he never got mad at, like Fred said, he got mad at things and he would leave.
Right.
No, these little things, of course, there's a lot of rules in government.
Yeah.
And they've been getting worse. But I don't know, I take an attitude that it's the big things that count.
It's a good attitude. That's why you've been here the whole time.
Yeah.
And I think that's fine. It's obviously different for everyone. You ever think about you had a long career as a scientist, but you ever think about what you would have done if you hadn't done research?
I probably would have been a medical doctor.
A pediatrician, right? Yeah.
Boy, I would have been quite different.
The whole field would have been different, right?
Yes, absolutely.
As an outsider, I say that,
I'm
but
sure I would have been retired by now.
Maybe. You're never going to retire, I take it.
Well, yeah, it was a funny thing when I was in the commissioned service, public health service. So I was there for almost 25 years.
Yeah.
But there, after you're there more than 20, you're supposed to be out.
Yeah.
Right. So I came back to NIH and I met with the HR personnel, and she's advising me whether to use the old system for retirement or the new one. Mm-hmm. And I was eligible for the old, and she said, "Oh, you should take the old." And I said, "No, I'm going to take the new one." She said, "Yeah, but you get much more when you retire with the old one." I said, "Yes, but you have to retire in order to get that advantage, and I don't have a plan to retire." So I took the new one. Of course, they took less money out of mine.
You make a really important point, Vincent, that had Bernie done something else, pox virology and a lot of other science influenced by pox virology would have been completely different. Bernie's been the undisputed leader.
You see the real Poxdoc?
For decades. Poxdoc, the real Poxdoc.
Excellent. Bernie, if you could go to dinner with anyone, who would it be?
A living or dead?
With you and Rich.
We went last night.
Yes, we did that last night.
Yeah, you're right. Okay. Rich, anything else?
No. That's good. This has been wonderful. I've been looking forward to this for a long time. You can find show notes for this episode at microbe.tv/TWiV. Send your questions and comments, TWiV@microbe.tv. Please subscribe on your phone or tablet using your podcast app. We'd like you to get every episode as they're released. It's free. Nothing that prevents you from doing that. If you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute and give us a few bucks a month, help with our expenses. Our guest today from the NIH National Institutes of Health here in Bethesda, Maryland, the real Poxdoc, Bernie Moss. Thanks so much.
Thank you.
We appreciate your time, and it was great. I really enjoyed that.
Yes, good.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing, Vincent. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. Thank you to ASV and ASM for your support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.